So we're going to get started on our kind of next session of the panels, and this is about a day and a half that we're going to spend focused on wildlife and threatened and endangered species. We're having to adjust the schedule a little bit because of the timing this morning, and we have a couple of consultants who are going to be making some of the presentations, and they're coming down from Fairbanks, and they won't be here till around 2 o'clock. So we're waiting for them, and we're also making an adjustment because we have when the Keystone Center was in the process of looking for panel members, we found a panel member who we thought would be a valuable asset to the process, who is a wildlife toxicologist and worked for U.S. Geological Survey. And Pebble has done some toxicological analysis in the environmental baseline document on what we're calling trace elements. But we don't have the individual here because of it was short notice on our part, and so the consultant who did the study and who would be making the presentation, Mark Stelljes, is actually not here. So what we're doing instead, and we apologize, that was our fault for not putting that on the agenda sooner than we did. But so what we're planning on doing is actually showing Mark's presentation from the agency briefings that Pebble did back in February of 2012. And we're just going to do that on the trace elements. And this is Mark Stelljes actually giving the presentation. Mark has a PhD in toxicology, and we don't have his bio actually in our information, but we can get that to you. So it's about a 40-minute presentation. And it's very similar to the presentations that you all have seen, except there won't be anybody standing here. You will actually see Mark on the screen in a couple of places. So we'll do that. You'll have that context for the discussion as we get into it. So without further ado, let me go ahead and start that. I think the sound is all. We also made some adjustments to the sound quality. And some of the technical difficulties we were having this morning. So hopefully people can hear us, not only in the audience, but on the web screen. We're going to ask the presenters when we get around to that to make sure that you're not too close to the mic because we're getting distortion, but aren't not too far away from it so that people at home can hear. But please let us know at home if you can't hear. So let me go ahead and start this, and then we'll be back on track.
The goal is to establish what are baseline concentrations of naturally occurring constituents in those media, and what's the variability of those chemicals in those various compartments. So what did we look at? In soil, we looked at ammonia, chloride, cyanide, fluoride, sulfate, mercury, and then 26 metals in soil and sediment and plants, 11 metals in fish. And then as we did for surface water, we had a few samples that we took early on and evaluated for pesticides, polychlorinated biphenyls or PCBs, and did that just in the fish tissues to make sure they weren't getting anything in from outside the area. And we also looked at organic carbon. So we've been started sampling all of those media in 2004. That was done by CH2M Hill, and then in 2005, after we got involved with the trace elements program, we modified the sampling locations and densities to be proportional to habitat abundance, as I mentioned before, and that enabled us to have a pretty good picture of the types of plant samples, the types of plants that were prevalent, and collecting as many of those types of plants as we could and analyzing them, rather than just doing a random grid, where then you may have 2 samples in a certain landform and you don't have enough data to be able to evaluate that landform, you have to go back out and collect additional samples from that landform. So we designed the program to make sure that we were collecting enough samples from each of the different landform types with superficial geology to have a pretty good understanding of what's in the soil and what's in the plants around. We sampled the plants twice per season. We sampled the vegetative growth early in the summer, and then we sampled again in the late summer where the berries were produced. And we did that because, of course, there's differential uptake with many chemicals between vegetative tissues and reproductive tissues. And also, they're eaten by different things, and so you want to know what the concentrations are in the various parts of the plants. And then we also evaluated aquatic plants as part of the pond program. Sampling locations. Every one of those multi-tiered rows is a sample location. The more of these little lines that you see, these are actually 5 different sample locations listed here. It's because we collected 5 different media there. So we collected sediment, aquatic plants, terrestrial plants, soil, and sediment, and analyzed those in that one sample. Other places you'll see there's just one line, and those are soil samples only. Most of the places we have soil and vegetative samples co-located. So every place we collected a plant sample, we also collected a soil sample. This is Steve Krupe back in the day. I think this is kind of becoming a annual photo at this point. This is how we collected the shallow soil samples. You just pull back the tundra mat, and then collect the tundra material underneath the mat, and goes into a stainless steel bowl and gets composited and put into jars. The program evolution, we added Pebble East to the area in 2006 after that was identified as part of the deposit area. And we stopped sampling subsurface soil after the first season because the data told us that the concentrations at depth were all lower or the same as they were at the surface, and so we focused on just the superficial soils in the future. And then for aquatic plants and pond samples, we expanded in 2006 to look at 3 different types of ponds. You heard yesterday about 2 different types of ponds, the ones that are groundwater fed and are basically present year-round and have a consistent vegetative border, and the precipitation-fed ponds like this one that dries up, and most of this is already dried out in the summer. And then there's also ponds that are impacted by beavers, and so that prevents inlets and outlets. So these are ones that may have inlets or outlets, but they're being dammed up. So we looked at all 3 of those types of ponds. And we did all this work by helicopter. Unlike all of the poor SLR people that have to do all this stuff in the wintertime, I was fortunate enough, since I had to look at vegetation, I had to be out there when there actually were plants on the ground, so I got to be out there in the nice summer and do this work, although then we have the bugs. Sample locations of the transportation corridor, we also collected samples and evaluated those for soil and plant material and some sediment. These are the locations along the route. Here's the general deposit area, and this is the divide between Cook Inlet and Bristol Bay drainages. Those were done in 2004 and 2005. So let's talk a little bit about the mine study area soil sampling. As I said, we initially based that on a random grid across the site and updated it to be proportional to the upland habitats. We had 117 locations sampled for soil. Some of those were, all of those were sampled once. Mine study area plant sampling. We collected over 50 different plant species and had them analyzed for trace element concentrations, including each of these groups: trees, shrubs. Trees out there are pretty hard to find. Shrubs, forbs, moss, lichens, grasses, and sedges. As I said, we sampled each of those twice each summer to make sure that and then we went back out a second year to make sure we had 2 sets of data for each location. So we were able to compare from year to year that way. And we sampled 70 locations. So as you recall from the soil, we had 117 soil sample locations, and 70 of those also included plant samples. Now, this is a kind of a busy slide, but it provides a summary of all the sample locations and the number of samples that were collected by year for sediment. And so we had anywhere from 14 to 50 samples of sediment from the 3 main rivers. We had anywhere from 12 to 22 samples from 6 to 21 ponds, and we had 9 seeps with 12 sediment samples that we collected in 2005. And then in 2004, CH2 also collected some samples from basically standing water in upland areas. They didn't qualify as ponds and they're not rivers, so I'm just calling them standing water. And those had quite a different pattern from the sediment there, and the soil there had quite different patterns from most of the rest of the sediments. Overall, we had 197 total sediment samples that we analyzed. And this breaks down the sediment sampling by drainage and by type. So we had 12 seep, 55 pond samples, and then pretty even distribution here amongst the 3 different rivers. For fish, sampling was conducted as part of the fisheries study, collected 5 species, all the ones that were available up there to collect. Sampled 4 lakes: Big Wiggly, Frying Pan Lake, Black Lake, and Lake 2. And sampled 4 rivers, including Cascanet Creek, in addition to the 3 that we've been talking about. Overall, we had 366 whole body analyses of fish from rivers. And then we did mussel-only analyses on 55 samples, just to compare total to mussel. And then in lake fish samples, rather than analyzing the whole fish, we analyzed the mussel in all the samples and the liver in addition in about half of them. So this breaks the fish sampling down by the same way that I did for sediment. 162, about half of the samples almost came from lakes, and very few came from Cascanet Creek, and most of them came from the rivers of interest, or the rivers of primary interest, I guess. So plant sampling summary, what did we collect for plants? Well, we collected 4 different species of trees. Most of what we collected were shrubs, over 300 vegetative samples and 80 berry samples. Those are the little willows and things like that that are all over the place up there. Forbs, which are essentially flowering non-woody plants, 146 samples there. Grasses and sedges, mosses and lichens. The point here is that we have a pretty robust data set for pretty much all the different types of vegetation except trees, and there are very few trees in the study area. At least anything taller than me. Very, very rare to find out there. Why did we sample so many different species? Well, primarily, it's because we want to make sure that anything that anyone or anything might consume, we have some understanding of baseline conditions for. Because there's a lot of different medicinal and traditional uses of many of these plants up in the area. By the Native Alaskans, we want to make sure that we know what's in those plants. We want to know their baseline. Multiple food source for wildlife. We want to know what's in the berries so that when the grizzly bears are eating them, we know what their baseline levels of exposure are going to be. And it also, down the road, improves our confidence in selecting indicator species. Like, do we want to sample a moss consistently over time as an indicator of potential contamination? This kind of information can help identify which are the most appropriate surrogate or indicator species to follow through the project. This I'm not expecting you to read, but it's an example of the types and diversity of the plants that are used by the native Alaskans up there. And you can see of this list, we sampled quite a few of them. All the ones that were at Pebble, we sampled. This is a full list of all the ones that we know that they use up in that general area. So the summary of the sampling by medium by year is on this slide. So the majority of our samples were split up pretty evenly between 04 and 05 for the soils, and then as we went on in the plant program, most of our plant samples were in subsequent years. Lots of samples. 253 soil samples, almost all of them are surface. The elements were generally consistent in concentrations at depth and at the surface, as I mentioned before, which is why we stopped doing the samples at depth. Several metals and ions were present, up to 3 times higher in the top foot than in the 2 feet below that. And that was enough to tell us that we should just focus on the superficial samples. Results for soil sampling. Those talus slopes that were talked about yesterday, all the crumbly, gravelly rock that's been broken up and there's essentially no vegetation on it, had the highest concentrations of more than half of the metals across all the geological landforms that we sampled. The concentrations were highest in moraine type samples for the rest of the metals. And that was particularly evident for barium. It was quite higher in moraine samples than in other types of geology. And I mentioned cyanide in surface water, and here it is again in soil. We actually have some cyanide that shows up on these talus slope samples. And again, I think it's because of the presence of cyanogenic bacteria in the superficial soils. So if I knew how to handle PowerPoint like you did, then I could actually not have you get overwhelmed by all these words at once. But anyway, here you go. I'll go through these key soil findings. The concentrations were quite diverse across the site, as you would imagine. They also vary within a specific location from sample to sample. The concentrations are influenced by the landform type and the habitat, but we weren't able to identify any consistent or significant correlations between the habitat type, the landform, and the concentration of really any of the trace elements. Aluminum and iron were the most abundant, as you would expect. We did find biogenic sources of petroleum range hydrocarbons in the shallow soil in all of 11 of the samples that we evaluated, up to almost 1,300 parts per million of residual range organic petroleum hydrocarbons. Now, these are not residual range organics from a petroleum spill. These are residual range organics that represent biogenic material that is broken down. And if you look at a gas chromatogram of this stuff, it's very different looking from residual range organics. You can very easily tell that this is not a petroleum-based source. It's a biological-based source. So it's very interesting that you have up to almost 1,300 parts per million of hydrocarbons that elute in the range that's identified as residual range organics. Similarly, with diesel range organics, we also have a highest concentration of 137 parts per million there. So when we get to many years down the road and we're looking at, say, for example, a petroleum release, if we find DRO and RRO, that doesn't necessarily mean there's been a release. because we have to make sure that we look at the gas chromatogram and understand, is this a biogenic source or is this a petroleum source? Then we also found cyanide in soil samples, and not just once or twice, but 84% of the time. So this is not just an isolated incident. Concentrations are quite low, 0.2 parts per million. These are well below any level of concern, but it's interesting that that cyanide is being formed in the environment. Now, Hugh showed you some box plots in groundwater, and I'm going to show you one now that's a little bit different in how it's set up than the way his was set up. Still the same basic box here, with the median here is a little square instead of a line. And then we have the 25 at 75, 5, and 95 percentiles. And then these are all basically outliers that fall outside of that distribution. This is copper, as you might expect. When you look at all of the soil samples collected for copper, a lot of them fall within a fairly narrow range. But then when you get into the samples closer to the deposit, and you're starting to see some substantially higher copper concentrations. With molybdenum, you have a very, very narrow range of concentrations, but you have these same kind of outliers from the samples near the deposit area that you get for copper. You don't see that same kind of range of outlier high concentrations for the ones that are less associated with the ore deposit. Plant sampling results. We collected a lot of samples of bog blueberry, and these are the most common species that we collected. They're also probably the more common species out there. So we're collecting the samples consistent with their relative abundance. And these are quite robust sample sizes for plant analyses to enable us to do statistics. Key plant findings: we found different elemental concentrations across different plant species. And generally, concentrations were lower in berries than in the leafy tissues. They were significantly lower in berries than in vegetative tissues for these 7 metals: aluminum, barium, calcium, cobalt, magnesium, manganese, and zinc. We did find that concentrations were generally higher in mosses and lichens than in other plants, particularly green terrestrial moss, and which had much, much higher concentrations of several of the trace elements. And even though we saw these higher concentrations of trace elements in soils in the talus slopes primarily, we did not see that same kind of thing in plants, so that the areas where the elements were high were not necessarily the areas where the plant concentrations were high. And then cyanide is also present in 36% of all the plant samples and 25% of all the fruit tissue samples. This is an example of the primary species that we collected that we had enough samples for to do statistics. This is dwarf birch, short stalk sedge, reindeer lichen, crowberry, narrowleaf Labrador tea, moss as a group, diamond leaf willow, bog blueberry, field horsetail. And these are July and August for each of the 3 sampling events that we had for sodium. And when you look at this, it's pretty clear that moss stands out. The concentrations are here, and in every sampling event, the concentration was highest in moss than in these other species. But you can also see quite a variability in both seasonally, short stalk sedge in July and short stalk sedge in August, being a little purple bar behind it. And you can see it from species to species, where the dwarf birch, you're not getting much sodium, but you're getting much more in the sedge. If you're thinking that it's a shrub, so it's not taking it up much, then you go over here to another shrub and it's taking it up more. So very species specific. Aluminum in vegetative plant tissues shows the same kind of thing. Higher in mosses and reindeer lichen. Here's the reindeer lichen for sodium. One of the higher ones, but certainly not unique for sodium. But for aluminum, it's very clear that these two, reindeer lichen and mosses, stand out. So let's take a little bit of time to look at the sediment sampling results. 197 samples. We analyzed 34 of those for acid volatile sulfide simultaneously extracted metals, or AVS-SEM. Some of you may be familiar with that term, to evaluate bioavailability. That's looking at is there enough material in the sediment to bind up the metals so that they're not available to organisms. And this gives you an indicator of that. The elemental abundance was similar to soil, as you'd expect. Key sediment findings: each of the major drainages have different signatures. So that includes those little minor drainages that I mentioned, the standing water areas. Anions and cations were generally much higher in the standing water areas, ponds and minor drainages, than in flowing waters. And sort of expect that. And seeps, however, had lower concentrations of cyanide than other types of samples. And I think this supports the cyanogenic bacteria hypothesis, because the seeps are essentially an expression of groundwater. You're not probably going to have bacteria in the groundwater producing cyanide, so you're not seeing really any until it gets out into the surface environment. Zinc in sediments as an indicator of how things look across the different types of water bodies. South Fork Cocktuli had the highest mean concentration of zinc. All of these bars are average concentrations. But otherwise, it's pretty consistent. Not a factor of 10, 15% difference across all these different units, but double in the South Fork. You look at chloride, you have a different pattern. Here it's higher, as I mentioned, in the standing water areas, the ponds and the minor drainages, and areas where you have flow, you have lower concentrations. Cyanide, summarizing what I had just mentioned, that seeps have almost no cyanide in them, but and ponds up to 0.7 parts per million, and minor drainages about the same. Flowing systems a bit lower. Now we'll turn to fish and look at the whole body results. We analyzed Olivardin, Arctic grayling, and coho and Chinook salmon, and copper and zinc are present at the highest mean concentrations in all three main river systems in the fish tissues. Nickel, chromium, and lead were substantially higher in fish sampled from the North Fork Cocktuli than from the South Fork Cocktuli. Cadmium was substantially higher in South Fork Cocktuli fish than in either of the other two fish in the other two drainages. Mercury was significantly lower in South Fork Cocktuli samples than in the other two media. So this demonstrates that there are chemical-specific and drainage-specific differences in trace element concentrations. And there's also a species-specific statistical difference within a water body for most elements, so that the Arctic grayling and the Chinook salmon would have significantly different concentrations of the same elements. Here's an example looking at the North Fork, looking at 2004, 2005, and 2004 and 5 combined in red. North Fork, South Fork, Upper Tulare, and Cascanet Creek are the four bars on each section of this graph. This is the mean concentration of copper in whole fish, and it's fairly consistent. You see a slightly higher concentration of copper in fish from Cascanet Creek in 2004, but it's lower in 2005. So overall, I'd say that copper in whole fish samples from all three drainages are essentially the same. If you look at nickel, in whole fish samples. What you're seeing here is a substantial difference in the North Fork versus the other drainages. The North Fork, you're seeing about threefold higher concentration average in of nickel versus the other three drainages. Looking at cadmium, in the same setup here, you're seeing a higher concentration in the South Fork. So depending on the element, you're going to have higher concentrations in different fish. Now looking at the liver and muscle results, here we often saw similar concentrations between liver and muscle, but where they were different, liver was almost always higher in concentration than muscle, which is what you'd expect, since I call the liver the landfill of the body. It really processes all of the bad stuff, and so you'd expect that all of the metals that end up in your body are going to go through your liver, and not surprisingly, there's higher concentrations in the liver than in the muscle tissue. And that's quite a bit higher, up to 188 times higher in the liver tissue than in muscle tissues for these four compounds: cadmium, copper, molybdenum, and silver. And most of the time, I would say the livers are not typically consumed. If you're going to use a fillet or something, but we analyze the whole fish and these separately in case you eat the whole fish or you're just going to eat fillets. Right, so that way we can look at potential current background risks and any potential future risks based on the type of food that's being consumed within the fish. If we look at copper in liver and muscle tissues, this is an example of what I was saying, another box plot here. So liver, we have a pretty wide range of concentrations, and it comes up to almost 100 parts per million on the 75th percentile, and gets up to almost 400 parts per million in liver tissues. But muscle, you could see on this scale, doesn't even really show up. So almost no copper in the muscles relative to liver. So to sum up, we collected approximately 1,765 samples from soil sediment, plants, berries, and fish in a 5-year period, and adequately characterized all of those media, I believe, for baseline conditions for trace elements and naturally occurring constituents. And different drainages have different patterns of trace elements, and landform and habitat and species all differ in elemental patterns. And so it's a complex system out there. The information that's in this EBD for trace elements is really designed to provide a foundation to build on with determining what to potentially monitor through time, where to do the monitoring, and how to make sure that we're going to identify anything that happens well before any concern levels get reached, because we know what the baseline conditions are. We can go out and we're going to know if something changes. Okay, any questions?
Thank you, Mark. I hope that was useful, and unfortunately Mark is not here and can't answer questions, and so we're going to have to be cognizant of that as we move forward with this discussion, because unfortunately we can't bring Mark up to answer questions. Our panel members can't. We'll be able to review the chapters, and this is a good setting, good context for that discussion. And we apologize that we can't have a more engaging discussion with Mark since he's not here. So we are still waiting on one of the consultants, or actually two of the consultants whose plane landed about 5 minutes ago, and they're on their way over here. One of the consultants for this afternoon's discussion is actually here.